In this video, we will discuss the steps of glycolysis. Remember, we previously learned that in photosynthesis, which occurs in the chloroplast, oxygen and glucose is produced during the light and the dark reactions. And then those two products exit the chloroplast to go to the cytoplasm of the plant cell. We also learned that a plant cell can undergo cellular respiration because the oxygen and the glucose can be used in that process. The glucose will be transformed first into a molecule that is smaller, and it will cross up into the mitochondrion across the inner and outer membranes and eventually reach the matrix, so that all the steps of the cellular respiration process can complete. So let's look at what happens here in the first step as glucose is transformed into a molecule of glucose 6-phosphate. The phosphate group is attached to carbon 6 and how this happens is by one ATP coming in here and giving off a phosphate group. It leaves as adenosine diphosphate. Next up, we'll see that glucose 6-phosphate will be transformed into fructose 6-phosphate. It is still a carbon 6 molecule, and it still has that phosphate group attached to carbon 6. Now, in this simplified video discussion, I am not going to look at all the enzyme names, all the enzymes that are required in this process. We're just looking at the basic steps in this video. After fructose 6-phosphate has formed, we will see the formation of a fructose 1,6-bisphosphate molecule. So that bisphosphate name tells us that there is a phosphate group attached to carbon 1 and to carbon 6. Again, here we need some ATP input. And a molecule of adenosine diphosphate will leave. The phosphate is taken from the ATP and attached to the fructose 6-phosphate to make fructose 1,6-bisphosphate. The next step involves the formation of two molecules. The first one is dihydroxyacetone phosphate. And the other one is glyceraldehyde 3-phosphate. There's an enzyme that is taking this 6-carbon big fructose 1,6-bisphosphate and changing it into two smaller 3-carbon molecules. Glyceraldehyde 3-phosphate is a 3-carbon molecule, and so is dihydroxyacetone phosphate. Now, to simplify things here at this point, we're only going to look at what happens here next with the glyceraldehyde 3-phosphate molecule. But what I can tell you is that depending on what is needed in the cell, at any moment in time, the dihydroxyacetone, in fact, can be changed into glyceraldehyde 3-phosphate. And this is a reversible reaction. There's an enzyme that does the catalysis here, like I said, based upon how much glyceraldehyde 3-phosphate is needed. If the cell needs to make more ATP, then it will be changed into glyceraldehyde 3-phosphate. The glyceraldehyde 3-phosphate undergoes something interesting next. At this point, as we now can look at this transformation of the dihydroxyacetone phosphate to glyceraldehyde phosphate, we see that, of course, there can be 2 molecules of glyceraldehyde phosphate present if some of the DHAP is converted. So what we'll do is we will write down the next steps on one side here. We don't have to repeat it on the other side. We can just put a big ×2 over here because these reactions will be the same as what we see on this side. So we're going to need some reducing power here, and that comes from an NAD plus molecule coming in and picking up some electrons to be reduced to NADH. And this will form for us a molecule of 1,3-bisphosphoglycerate. Of course, there's also an enzyme involved here. This molecule of bisphosphoglycerate will be changed in the next step into a molecule of 3-phosphoglycerate. And here we see for the first time the production of 1 molecule of ATP. So what happened here is that adenosine diphosphate came in and it picked up a phosphate from the 1,3-bisphosphoglycerate. Remember, this was a molecule consisting of 2 phosphate groups. Bisphospho refers to 2 phosphate groups. So if we take away 1 phosphate, the molecule of 3-phosphoglycerate now has 1 phosphate remaining within it. Next, 3-phosphoglycerate will be changed into 2-phosphoglycerate. And after that, 2-phosphoglycerate will be changed into phosphoenolpyruvate. Now, during this step, 1 water molecule does exit here. It is a dehydration. And lastly, the phosphoenolpyruvate will be changed into pyruvate with the help of an enzyme. And at this point, we again see the production of an ATP molecule, showing us that the last phosphate is removed from our phosphoenolpyruvate, as there is no phosphates left in a molecule of pyruvate. Now, pyruvate is still 3 carbons big, just as all these molecules in this sequence here ahead of it. And it will go next to the prep reaction of cellular respiration. To summarize, it's always a good idea to write down what is our input and our output. And we will do this in the form of net output. We do input and net output. So let's look at the reactions together here, and then you'll see that we have an input of 2 ATP and we have an output of 4 ATP. We have 2 ATPs leaving down here, one from the bisphosphoglycerate and one from phosphoenol pyruvate. But remember, we have to multiply that by 2. So 4 minus 2 gives us 2. Our net output is 2. So in order to make 4, we had to put in 2. That's why we say our net output is 2. We did put in 1 glucose molecule and we're going to end with 2 pyruvate molecules. We see 2 water molecules left or exited, and then we also see 2 molecules of NADH. Remembering that everything down at the bottom needs to be multiplied with 2. So we'll discuss the prep reaction and the citric acid cycle or Krebs cycle in the next video.